Welcome, everybody. So excited for another webinar through our tumor section. On behalf of the AANS and our tumor section, this is a new initiative that we're really excited about. It's focusing a little bit more on research and journal club type format that we're going to have residents and other types of research fellows present. I do want to thank again the AANS for their help in organizing this. I also want to acknowledge Dr. Joseph Ellenbogen's help from the University of Toronto, Dr. Geleray-Zadeh and Dan Cahill, our co-chairs for the research subcommittee of the tumor section. I'm going to let Dr. Ellenbogen take it from here, but once again, thank you everyone for joining. Really excited with this new webinar that we're launching today.
Wonderful, thank you. Yeah, so as Dr. Hadjipanayas mentioned, this is we're sort of starting a webinar series that's called the Limelight Webinar Series, and it's meant to highlight some of the trainee and early career investigator research through the tumor section. So this is the first session, so thank you all for attending, and thanks to the panelists who I'll introduce as we go through them. The faculty discussant that we have for today is Dr. Ian Parney. And so he just joined, I believe. Dr. Parney, I see you there. Okay, fantastic. So
yes, I'm here. Sorry to be a couple minutes late.
No worries. I'm sure it's a busy day. So we were just talking about the structure of the webinar. Maybe I'll start with a brief introduction about Dr. Parney, and then he'll discuss a little bit about his research works before we go into the trainee presentations. So Dr. Ian Parney is a neurosurgeon at the Mayo Clinic in Rochester, where he's the professor and vice chair of research at the Department of Neurologic Surgery. He's a clinician scientist with a clinical practice and lab focused on malignant brain tumor, and his lab research is brain tumor mediated immunosuppression and liquid biopsy. So we're very fortunate to have him as our first faculty discussant. We're very thankful, and so I'll pass it over to him. You should be able to share your screen if you'd like, or you can just, floor is yours.
Well, thanks very much, and so nice to see everybody here virtually, and thanks for those of you who are presenting. This is really quite an exciting thing. Is everybody seeing this, the full slide mode now? Yeah. So I don't want to talk too much because I think this is meant to be about the presentations from trainees, but just to kind of give you a little bit of an idea of who I am and how I ended up as a brain tumor oncologist and what I do, neurosurgical oncologist, we can just kind of go through things. So this is, those of you who've ever seen me give a talk, you know that this is one of my favorite paintings. This is in the Prado in Madrid. And you see this very confident looking neurosurgeon taking out the stone of madness, inspiring confidence in families around. So we're a little bit better than this now when we do surgery, I'd like to think. But we've still got lots of room to grow. And that's really all about research, which is what we want to talk about today. I titled this the peripatetic neurosurgeon because I've been around a little bit. I'm actually from Calgary in Western Canada. Grew up there, and then I went to college at McGill University. This is actually a picture of the backside of the Montreal Neurologic Institute, home of Wilder Penfield. And I lived within about 3 blocks of the MNI for the entire time I was in college. I went from McGill back to Alberta in Western Canada to Edmonton, to the University of Alberta, which was very, very cold. And did my residency, graduate school, and was briefly on faculty there. I then went and did fellowship at UCSF with Mitch Berger and others in neurosurgical oncology. Moved back to Calgary, and then from there moved to the Mayo Clinic, where I've been now for almost 17 years. So just sort of a little background of where I've been and how I got to this position. In terms of the research things that I do, I say you have to have a Venn diagram. And these are kind of the things that I have focused on in my laboratory research, so immunosuppression with gliomas, and in particular immunosuppressive monocytes, extracellular vesicles, and various overlaps of this. Within that, and overlapping that, I would say I have an interest in clinical trials. And then on top of that, the overarching thing is I'm interested in neurosurgical oncology outcomes. I probably should focus this. When I looked at this, I said, "Well, maybe my chair will tell me I need to fix on one," but we'll see. Um, so, what do I mean by all this? What have I done over the years? So, I started out really focused on glioma-mediated immunosuppression, various mediators of this. And this is a review article, a figure from a review article that we published a couple of years ago. But you can see these papers are kind of back when I started out. This was actually a big part of my PhD thesis, this paper. And so we're sort of interested in how these tumors shut down the immune system, because they're very effective at doing that both within the tumor and systemically. That interest in immunosuppression really got honed and focused over about a decade or so on these immunosuppressive monocytes. And there's various versions of that: myeloid-derived suppressor cells, for which there's various flavors, non-classical monocytes, other tumor-associated monocytes, or macrophages. And so we published a number of papers related to this over the years. That interest in immunosuppression led to an interest in extracellular vesicles, these little tiny bits of cell that get shed from really every cell in the body, but they're shed in abundance from glioma cells and other cancers. And we got interested in them because we thought, well, maybe this is a mechanism by way or how gliomas will induce immunosuppression. And it is. We can show that these glioblastoma EVs will induce immunosuppressive changes in monocytes. They become myeloid-derived suppressor cells and non-classical monocytes when they're exposed to GBM EVs. And they do that through PD-L1 and IDO1, amongst other mechanisms. But that has also led to an interest in extracellular vesicles as a source of biomarkers for liquid biopsy. And that's actually a big part of what I do in my research at the moment, is plasma extracellular vesicles. We're still on the hunt for a reliable way to identify tumor EVs in plasma. You can find them. They're just rare. And so we're working at sort of trying to magnify that signal. But what we have found, which is really very interesting, is that the other EVs, the non-neoplastic EVs in plasma, are wildly deranged in GBM patients systemically compared to normal donors or even other brain tumor patients. And so that's a big part of what my work is focused on at the moment. As I said, I have an interest in clinical trials. This is sort of moving from thought to action, and some of these are published, some of these are ongoing. This is a series of things that I've been involved in as either PI or co-PI over the years, different vaccine trials and checkpoint inhibitors and cytokines. And I'm sort of the accidental oncovirologist by being in the right place at the right time. I've been involved in a few oncolytic virus studies, though it's never been a huge focus of my career. And then a big interest in neurosurgical oncology outcomes, dating back to when I was a fellow working with the Glioma Outcomes Project, but just sort of really trying to learn from our own cases how people do when we do surgery for them. And this has actually been a big push with our group here. I have a clinical neurosurgical oncology research group, a bunch of residents, fellows, and medical students, including actually Cecile Riviere-Cazot, who's on the call here today as part of that. And so we've been able to go through the about 4,000 brain tumor procedures that I've done since coming to Mayo Clinic, and we are really plumbing that for a lot of data on how people do with tumors, intraaxial tumors in different locations in the brain. So I'll stop there because I said I think this is meant to be about everybody else, not about me. And it's just sort of a brief overview of some of the things that I've been involved with in neurosurgical oncology. But I'm really excited about hearing everybody else's presentation. So.
Wonderful, thank you. I mean, it's great to hear sort of the wide breadth, and you get a a flavor of all the different areas that you're working in, which is super inspiring and interesting for all of us to see, especially early in our training at various stages. So we'll go next to our first trainee presentation. Darsha, do you want to share your screen while I just introduce you? So Darsha Malla studied biomedical engineering at Johns Hopkins, and now he's in his MD PhD in his final year at the University of Maryland School of Medicine under the supervision of Dr. Graham Woodworth. He'll be talking about an advanced murine brain tumor resection platform to model and study residual invasive disease in glioblastoma.
Great. Thank you for that introduction. Can you hear me and see my screen okay?
Yes.
Great. Awesome. All right. Well, good afternoon, everyone. Thank you to the organizers again for the opportunity to present. My name is Adarsha, and I'm an MSTP student at the University of Maryland School of Medicine. And today I'll be presenting on behalf of our team on our work towards developing a controlled stereotactic brain tumor resection platform to model and study residual invasive disease in GBM. And so I won't spend too much time on this slide because I think we're all relatively familiar with glioblastoma or GBM. GBM is the most common primary malignant brain tumor in adults, and it affects around 15,000 new people in the United States each year. Despite aggressive multimodal standard of care treatment that includes maximal safe surgical resection, followed by chemotherapy and radiation therapy, these tumors almost universally recur and are fatal, with the average patient surviving around 15 months and only around 7% of patients making it past 5 years from diagnosis. And so again, as we probably all know, GBMs are challenging to treat for a number of reasons. We just heard from Dr. Parney, they're highly immunosuppressive. They're also diffusely infiltrative and can harbor a significant amount of molecular and cellular heterogeneity. But what kind of piqued my interest when I joined the lab is this residual invasive disease component, which is composed of invasive cancer cells that have infiltrated past the tumor margin and are inaccessible by surgery, and seem to really be kind of the primary driver of near universal tumor recurrence. And what made this residual invasive disease component even more interesting to me is that recent studies had demonstrated that core and margin regions can actually have profound differences in their tumor cell state and biology, as well as their immune cell composition, tissue biophysics, and vasculature and vascular structure. All evidence kind of pointing to the idea that therapeutic vulnerabilities identified in the tumor bulk might not actually apply to the invasive margin and the residual invasive disease. And so this kind of made it seem more important to try to study the residual invasive disease. But since we can't really study it well in humans, given that patients very rarely undergo repeat biopsy or repeat resection, I kind of looked at animal models, and it turns out some preclinical animal models do recapitulate the core margin differential biology that we see in human GBMs. But as you can probably appreciate from these histological images, the invasive margin and residual invasive disease biology really make up a small fraction of the total tumor mass. And so to me, the way to really get at this would be to try to go in and resect that tumor core. It turns out people have tried this, but with varying and overall kind of limited success. As you can probably imagine, doing resective surgeries in a mouse or rat is technically challenging, time-consuming. Animals generally don't do very well. There's high variability with kind of microdissection in a small surgical subject, and overall harsh handling of the tissue and residual microenvironment. So you can kind of see in this example, they were able to resect kind of partial bulk of the tumor. And then in this bottom example, they were able to remove most of the tumor, but there was some kind of disruption of the residual microenvironment. And they actually state in their methods that the surgeries were performed by experienced neurosurgeons. So kind of despite these considerations, I still wanted to give this a shot and see if we could really create a model that is enriched for residual invasive disease biology by going in and safely and reproducibly trying to resect out that tumor core. And fortuitously, around the time I was joining the lab, we were actually getting the NICO Myriad set up in our ORs at the hospital, which I'm sure many of you are aware of or have heard of this tool. It's the tool that was used in the ENRICH trial, and it's an endoscopic resection tool that has an integrated suction and collection mechanism. So kind of in a bedside back to bench effort, we worked with the NICO company to get a miniaturized version mounted on our animal stereotactic frame. And the way the device works is pretty neat. So there's a resection cannula that has an inner cylindrical blade that oscillates at around 1,400 times per minute. And so the tissue is suctioned in and resected and actually collected. And the device also has a dial that you can see here that you can rotate that allows you to control the direction of the resection aperture. And as a result, allows you to kind of control the direction of cutting. Another nice aspect of the tool is that it has a low profile design. So this approach is kind of much easier on the animals compared to conventional approaches that require significant bone removal to get access to the tool. So this is kind of an example where they had to do like a 4x4 millimeter craniectomy to kind of get tools and attempt to remove tumor. And so now that we had this tool, mounted and potentially usable for preclinical applications, we were interested in really trying to define the dynamic range and precision of this approach, kind of systematically by tuning the user-defined parameters. So using MRI-based volumetric quantification, we analyzed the volume of the resected tissue and developed different protocols that allowed us to attain a range of resected volumes, including a biopsy protocol, a 1x rotation protocol, and a 2x rotation protocol. The tool also comes in 2 different cannula sizes, so this enabled us to have almost a 10-fold increase compared to the smaller 23 gauge cannula size. So this 19 gauge is one that we use for many of the rat resections. And we were also interested in quantifying the precision of this MIS, this murine intracranial surgery approach, in resecting a targeted tissue volume. So we designed a prospective adaptive study to evaluate how well this approach could be used across 5 different preclinical GBM models. Based on quantification of tumor volume using pre- and post-operative MRI sequences, we obtained a median extent of resection using the 1x protocol of around 80% and using the 2x rotation protocol, median extent of resection around 98%. Additionally, we were interested in trying to mirror some multi-regional stereotactic biopsies that are often done in humans. Being able to do this in a mouse in a mouse tumor would be kind of neat, especially considering we could also have the capability for tissue collection. And so, I kind of targeted the core bulk of the tumor here and this invasive margin where invasive cells had infiltrated the corpus callosum, and we were able to, pretty effectively biopsy and collect that tissue. Next, to kind of further evaluate the collection abilities of the system, sterile collection abilities. This is kind of important given there are no tools or methods for accessing murine intracranial tissue in a kind of precise, controlled, and tunable manner while surviving the animal. And this, of course, limits longitudinal studies of biology or treatment responses. And so, with the larger volume resections, we resected more cells, and on average, the viability was 80 to 85%. Coming straight out of the container, the cells were pretty well dissociated. And when plated in a serum-free media, we could actually sterilely expand and freeze down and then re-expand the cancer cell and the neurosphere compartment of the resected tissue. Similarly, we explored whether we could also resect and analyze the immune cell compartment of the tumors, which currently this can only really be evaluated with euthanizing the animal and collecting tissue. And so, we were able to observe pretty nice T cell populations and myeloid cell populations. We also compared this to kind of conventional protocols that require you to sacrifice the animal and collect tissue and notice that the results were pretty comparable. These are just some representative MRIs and histologies from pre and post resection. So in the RCAS TVA genetically engineered rat model, you can see this T2 weighted hyperintense mass with some contrast enhancing volume that was completely resected, and the resection cavity is then filled with CSF. Same thing in the CT2A syngeneic model, complete resection of the bulk contrast enhancing mass. On H&E histology, there's very minimal bulk tissue, bulk cancer tissue there with, what's left behind is really this residual invasive disease component. Similarly, we tested our approach in 2 different patient-derived xenograft models, and we're able to achieve similar results with gross total or complete resection of the primary tumor bulk, with, again, really only the residual invasive disease left behind on histology. So armed with this new tool and approach, we were interested in exploring some aspects of residual invasive disease biology. So from longitudinal MRI imaging, we observed that the recurrence kinetics appeared to be related to the amount of residual tumor left after surgery. And so looking at this with linear regression across all of our models, we were able to kind of confirmed that extensive resection is actually associated with survival in these preclinical models. And then using a categorical Kaplan-Meier analysis, stratifying animals into greater than 90% extensive resection and less than 90% extensive resection, we really only saw the survival advantage in the patient-derived xenograft models. Next, we also noticed that the recurrent growth patterns of the model seem to be quite distinct. So in the syngeneic models, the tumors almost exclusively grew back into the cavity, versus in the genetically engineered models, as well as in the patient-derived xenograft models, we observed that recurrent tumors would oftentimes grow into the brain. And this is what we generally observed when we analyzed human GBM longitudinal imaging, is that the recurrent tumor oftentimes grew into the brain rather than just repopulating the cavity. And so we quantified the resection cavity volume over time in the preclinical models in order to be able to compare them to the human imaging data, and observe that really the HSR-GBM1 model and the RCAS-TVA model seem to best reflect the human GBM recurrent growth patterns. Finally, I'll just end with some molecular data that we collected. So we generated some matched primary and recurrent patient-derived xenograft samples by collecting tissue with the MIS approach, allowing the residual invasive disease to expand to a similar volume as the primary tumor, and then collecting that tissue again. And based on the GBM purity algorithm analysis, there was pretty similar tumor purity, which had a median of a 98.75%. And there was no significant difference between the primary and recurrent tissues, kind of ensuring a consistently high proportion of tumor-derived RNA across the samples. Based on dimensionality reduction and UMAP visualization of the samples, we actually saw divergence between the primary and recurrent samples. And this was further supported by unsupervised hierarchical clustering of the samples, where this analysis separated the grouping of primary and recurrent samples. To contextualize these transcriptional changes, we performed gene set enrichment analysis to identify the biological pathways that were differentially enriched between primary and RID recurrent tumors. Based on this analysis, we did see that RNA processing, metabolism, and DNA checkpoint pathways were enriched in primary tumors, whereas recurrent tumors actually exhibited an enrichment for neurodevelopment and synaptic signaling pathways. This is a bit of a busy figure, but just to kind of quickly summarize, we also did GSEA mapping with a human data set and observed that while there were a few different transcriptional programs upregulated across the data sets in the primary tumor, in the recurrent tumor data sets, they all converged on a shared transcriptional shift kind of for enrichment in neurogenesis, synaptic signaling, and nervous system development. And I think I'm a bit over time, so I'll quickly wrap up here, but we also looked at a few different classification systems and to see if resection alone actually changed the subtype classification. And based on the Garofano pathway-based classification, we did see enrichment of the neural subtype in our RID recurrent samples, which is observed to be enriched in rim samples when compared to core samples, as well as recurrent GBM when compared to primary GBM. Finally, based on this neuronal-like phenotype, we also looked at the expression of key glioma neuron interaction mediators, which are emerging kind of therapeutic targets in this emerging field of cancer neuroscience. And we did observe enrichment of neuroligin 3, BDNF, TRKB, and GRIA1 in our RID recurrent tumors, and we kind of confirmed at the protein level with immunofluorescence. So this is a summary and conclusions. We looked at extensive resection and found that it was associated with survival in our preclinical models. We also observed that the post-surgical growth kinetics and patterns varied significantly by model. So again, for something like developing cavity implantable therapeutics, potentially important to choose models carefully and choose models that grow into the brain in recurrence rather than simply grow into the cavity and are being washed by a cavity implantable therapeutic. We also looked at the molecular level and identified changes in the RID recurrent tumors that actually mirrored recurrent human GBM profiles. And overall, the goal was to try to create a post-surgical model of residual invasive disease. And both histologically and radiographically, we show that as well as at the molecular level with transcriptomics. And that's the end of mine, and I'm happy to take questions.
Great, very impressive work, Rasha, very elegant. I think we probably just have time for maybe one quick or two quick questions at most. I do have a question. Just are there certain types of pediatric tumors or of GBM models, mirroring tumors, that you tried where this just wasn't feasible? In other words, how much of this, your ability to do this, how much does that depend on the biology of the particular model that you tested?
Yeah, that's a great question. So actually, in our syngeneic and the genetically engineered models, we generally didn't have too many issues. When we got to the PDX models, we noticed that we had to incorporate some additional steps to ensure that after resection, there was kind of physiologic and complete hemostasis in the resection cavity. So we did have to incorporate some steps where we kind of rinsed and washed the cavity, irrigated the cavity with saline to ensure that there was complete hemostasis as those animals for some reason the nude mice that we use for the patient-derived xenograft models ended up oftentimes potentially bleeding a little bit after surgery and didn't end up doing well.
Right. Thanks.
Wonderful. I just maybe I'll open it up to the audience if there's any additional questions. I believe there should be an opportunity to either post in the chat or raise your hand and I can unmute. Maybe I'll just ask one quick question while we're waiting and then we can decide about other ones. So really impressive work. I think, having a preclinical model to study residual disease is quite needed. I'm curious about the logistics of it a little bit. So you touched upon hemostasis. Did you find, infection or sterility had any factor here, or was the recurrence so quickly or so quick in this model that wasn't really a factor? And also, is the tool that you got custom made? Is that something that is being made accessible? Is that something that can be purchased, or is it quite custom to your lab?
Yeah, so I'll answer your second question first because I think it's quicker, but this system is commercially available, kind of like the preclinical version of the Nico Myriad. And to your first question, we didn't actually have any cases of infection, and the recurrent or kind of the recurrence biology occurred, in like the syngeneic models over like 5 or 6 days, but actually in the PDXs, like the HSR-GBM1 model, these animals actually live for almost a month after resection. So there were kind of some difference in recurrence kinetics there that we think might also be a part of, kind of the growth patterns and the relationship between extent of resection and survival that we, kind of saw more strongly in the PDXs versus the syngeneic models. But yeah, long term, we actually didn't notice any issues with infection, kind of intracranially or with the skin once the animals kind of did well the day after surgery.
Okay, wonderful. Thank you. And thank you for your talk. So we'll go on to the next one. Sanjeev, are you able to share your screen? And while you do that, I'll introduce you. So Sanjeev Her is a fourth-year medical student at Drexel University College of Medicine, and he's currently completing his master's degree in Dr. Sean Harvey Jumper's lab at UCSF. He's also a combat medic in the Army National Guard, and he'll be applying to the match this upcoming fall. So keep an eye out.
Thank you. Are you able to see my screen?
Yeah.
Yep.
Okay. Excellent. Good afternoon, everyone. I'd like to thank you all for being here and thank the tumor section for the opportunity to present my work. My talk will be about the work I've done this past year in the lab of Dr. Sean Harvey Jumper, involving optical imaging-based random forest boosted prediction of recurrence in glioblastoma. So as we know and has been touched upon, glioblastoma is a universally fatal disease with a median overall survival of 17 months, and it's further complicated by inevitable recurrence with a median progression-free survival being 8 months. Extent of resection is one of the most predictive measures of recurrence, yet tumor remains in the form of microscopic infiltration. For 3 out of 4 patients, recurrence will happen along the resection cavity border or margins, but we have no idea who this will happen to or where it will recur, and so we can't treat it until it's already happened. And previous work has identified that infiltration of tumor cells is a major driver of recurrence for solid tumors. This is why I wanted to test the hypothesis that glioblastoma recurrence is largely driven by infiltration, and a machine learning informed measure of tumor infiltration, AI infiltration, at the resection margins can be used to predict focal recurrence. This study was a single center retrospective cohort study of patients greater than 18 years of age that had pathology confirmed IDH wild type glioblastoma who had intraoperative stimulated Raman histology performed at time of resection. Our training cohort had 60 patients with 273 samples, and our testing cohort 20 patients with 94 samples. Patient demographics and follow-up data were obtained via chart review. And the AI infiltration values were generated from FastGlioma, which was a recently published multi-center prospective study by Kandapuri et al. That leveraged stimulated Raman histology and AI to build a model for rapid quantification of tumor infiltration. So during resection, one sample is taken from the tumor core, and then following resection, as the margins are assessed, samples are also obtained. These unprocessed, fresh samples are then imaged using stimulated Raman histology to generate a pseudo H&E image that is analyzed at a pixel level and outputs an infiltration value from 0 to 1. Follow-up imaging for patients at time of recurrence is then cross-referenced with pre-op and post-op imaging to identify a site where a sample was taken at the margin as recurred or not recurred, with the examiner blinded to clinical results. And the median time to recurrence for our cohort was actually 5.5 months. So within our training cohort of 60 patients, 43 had recurred and 17 had not. The model that I built included clinical, radiographic, and molecular features, and significance testing between patients that recurred and those that did not recur showed no major significant differences in these features. So after checking for differences between the patient cohorts, I wanted to assess whether or not the sample distribution was also similar across patients. I first mapped each patient's samples in 3D space using their XYZ coordinates, along with spheres that were representative of the smallest, median, and largest tumor volume. So that way I could visually inspect for clustering of samples. And in this plot, each of the different colors and shapes represents one patient's samples. And so following that, I used pairwise Euclidean distance of samples within each patient to calculate their average standard deviation as a measure of variability in sample dispersion. And between the patients that recurred and those that did not, there was no significant difference in the degree of sample dispersion. And then to further assess the potential bias of recurrent samples being taken closer to the core and thus having an expected higher degree of infiltration, the distance for each margin sample to its respective core sample was calculated, and there was no significant difference between samples that recurred and those that did not. Next, I wanted to ensure that AI infiltration was comparable to the gold standard histopathologist-determined infiltration. So using various staining techniques, path infiltration was determined on a subset of 137 samples from 32 patients. The scoring was a 0 to 3 system, with 0 being normal brain and 3 being dense tumor infiltration. When grouped by path infiltration, pairwise testing demonstrated significant differences in the median AI infiltration across groups, except between 0 and 1, indicating that the AI-generated infiltration values correlated well with the histopathologist-determined infiltration. Following this, I wanted to identify a ground truth for the ability of infiltration to predict recurrence. I first assessed the degree of infiltration between core samples and margin samples using the path infiltration, which showed an expected increased infiltration in samples taken from the core. Following this, samples were split into those that recurred and those that did not, demonstrating a significant distribution of higher infiltration in the recurrent samples compared to the non-recurrent. These relationships were then tested using the AI infiltration, which again showed the same significant findings of increased infiltration in the core samples and an increased infiltration in recurrent samples. I then wanted to see if infiltration alone could predict focal recurrence. And as you can tell by these AUCs, it could not. But this led to me wanting to do further work to see if I could build a model that was predictive of focal recurrence. So the first step to building this model was identifying which variables to include. I did this through predictor screening of all patient clinical, radiographic, and molecular features. I did this using bootstrap forest partitioning to identify which variables contributed most to sample recurrence prediction. Among the top predictors were very well-known predictors such as methylation status and proliferation index, as well as the AI infiltration value. These variables were then used to screen classifier models using a 10-fold stratified internal cross-validation that I applied to logistic regression, k-nearest neighbors, support vector machines, random forest, gradient boosted trees, and extreme gradient boosting. Out of these different models that were screened, random forest demonstrated the best performance with a mean AUC of 0.85 and an AUC of 0.8 when applied to our test cohort. And amongst all of these models that were screened, AI infiltration remained one of the top 5 predictors of recurrence for 5 out of 6 of the models, and the #1 predictor in 4 out of 6 of the models. Next, I wanted to see how generalizable the random forest model could be made. To do this, I performed a reverse feature elimination using the same 10-fold cross-validation approach and using AUC thresholding of 0.8 as a marker for clinical utility. It showed that a model informed by only infiltration score, age, and extent of resection was able to predict sample recurrence with a mean AUC of 0.8. After determining whether a site would or would not recur, I wanted to see to what extent I could predict the actual site of recurrence. Given the propensity for recurrence at or near the resection margins, I determined if recurrence was within 5 millimeters, 10 millimeters, or was greater than 10 millimeters away from each sample. Logistic regression of each sample's infiltration score and their respective distance from recurrence demonstrated that an increase in AI infiltration resulted in an increased likelihood of recurrence within 5 millimeters and within 10 millimeters. To validate the spatial recurrence predictions, thresholds for recurrence were determined by identifying where the probability curve for a sample being greater than 10 millimeters from recurrence intersected with the probability curve of a sample being less than 10 millimeters or less than 5 millimeters from recurrence. These thresholds were then used to classify each sample in the separate prospective testing cohort as more or less likely to recur. Each classification was then compared to the sample's actual distance from recurrence with a greater accuracy in the 5 millimeter predictions. Following that, I wanted to visually represent how these predictions could be of use clinically. And so to do that, I plotted the tumor margin samples, and for each margin sample, the respective AI infiltration value, recurrence probability within 5 millimeters, and recurrence probability within 10 millimeters. And then I also plotted on here where the actual occurrence ended up happening. And so here I have pictured one from a patient in the training set and then one in the testing set. So I was able to demonstrate that stimulated Raman histology-based AI-generated infiltration values correlated with histopathologist-determined infiltration, and that the degree of infiltration largely contributed to focal recurrence prediction. This subsequently showed that AI infiltration can be leveraged for ultra-fast prediction of recurrence likelihood around the resection cavity. My hope is that these technologies will allow us to take current treatment paradigms that may have had some limited success and more precisely target areas that are most likely to occur, such as with dose escalation radiotherapy and catheter-based chemotherapy. I'd like to thank my mentor, Dr. Sean Harvey-Jumper, and his entire lab, as well as Dr. Holland in the Holland lab, and my thesis committee members, Dr. Karen Hurley and Dr. Edward Kim. And this work would not be possible without the wonderful patients and their caregivers.
And gee, that's very interesting data. Impressive that you can sort of do this and actually have this sort of shake out as a statistically viable finding. Can you sort of talk about how you might incorporate this into the operating room to make a clinical decision or into your radiation treatment planning you mentioned? I'm not sure if we're sort of there yet with this or if you need to refine it further, but how do you see this going forward in the future?
So I know that right now, it's just this is used in operating rooms just for research purposes, but it is very fast. As soon as a sample is put in, it's about a minute for each sample to determine whether or not it is considered still tumor, and it gives you that score as well. And so I think, that would be able to help guide resection in a sense. But then on top of that, I think knowing which sites are more likely to recur. I know there are a lot of dose escalation radiotherapy trials that kind of just broadly tested dose escalation radiotherapy, but I think, focusing more on these areas that are densely infiltrated might have a better effect
Right.
with fewer side effects.
And how about sort of correlation with other novel imaging technologies, be that, PET scans, not necessarily the standard FDG PET, but, FDOPA PET, or, UCSF has a long history of expertise with MR spectroscopy, multivoxel MR spectroscopy. Do your findings correlate?
So that's actually something hope to look into further in the future, because I think that, that would be really useful if we can, they do have some infiltration metrics that some of these imaging modalities generate, but I think being able to actually correlate that with, a lot of them don't have histopathology samples to correlate with. So I think in this sense, it would be very useful to be able to correlate those things so we could have more non-invasive techniques for measuring infiltration.
Thank you.
Wonderful. Thank you, Sanjeev. I think in the interest of time, we'll move to the next presenter, but then hopefully at the end, if there's additional questions, we can do them then. So our next presenter is Cecile. Are you able to share your screen?
Yes, I can.
I'll just introduce you as well. So Cecile, and I apologize if I mispronounce your name because my French is terrible. Riviere-Cazot is a fifth-year MD, PhD
It's a good
student
Canadian
at,
pronunciation. That was perfect.
It was perfect. Yeah.
So she's a fifth-year MD, PhD student at the Mayo Clinic in Rochester. She completed her undergraduate studies at UCLA. She is mentored by both Dr. Terry Burns and Dr. Ian Parney here, and her research focuses on liquid biopsy biomarkers in both the CSF and plasma for diagnosis and disease monitoring. So thank you for taking the time to come talk.
Of course. And thank you for the invitation. I'm really excited to get to talk about some of the work that we're doing at Mayo with our liquid biopsies, specifically related to proteomics. And I've somewhat cheekishly titled this talk, How to Learn from Your Mistakes, because this has been ongoing work for us for close to over 3 years now. In part, it's a very iterative process of thinking we're nearly there with the paper and then realizing that something's gone wrong and going back and learning every single time from our patients. But with that, I'm not going to give too much background on gliomas because I'm sure everybody is well aware of the problems that we're dealing with here. But I'll point to an example patient that's special to us because she's the first patient on our Maya reservoir trial for longitudinal CSF access. She's a patient that come with a recurrent GBM. Took her for a section. She already had recurred by the time she came for her post-op appointment, recurred even further after she went on lomustine, then progressed straight through bevacizumab. And so I'd argue that this is a somewhat expected and linear pathway through which most of our patients with gliomas progress, and it mirrors our current neuro-oncology pathway, where we are going from this discovery phase to translation and then sometimes application, rarely ever for GBM. But it's very linear, and there's very limited feedback as to where we're going wrong and how we're going wrong and how we can course correct. And so a big portion of the work that we've been doing here has been figuring out how we can develop this sort of alternative pathway that's iterative and guided by patient feedback, takes in candidate therapies and technologies that are emerging, tests them in patient, and then uses those data to iteratively refine and improve on what we can do for our patients. As part of that pathway, we need individualized biomarker feedback. And so that's where our work has really laid in over the past 3 to 4 years, and working to develop the toolkit that we have to be able to learn in real time from our patients. With that, we were really interested in glioma CSF proteomics. There's a number of couple of papers that have come out more recently. This really nice paper from Dr. Mansouri at Penn State and his group that show that you can use the CSF proteome to be able to distinguish one glioma subtype from another. We're really interested in CSF proteomics because I think it allows us both to potentially have monitoring, but then also pharmacodynamic feedback if you have a targeted therapy that you can then evaluate in CSF. But one of the things that is still very much unexplored in the field is, what variables do and don't matter. And we're in the business of rigorously developing our liquid biopsy biomarkers, and location is one of the things we have to consider. Because in the operating room, if we're obtaining CSF, we can get from a variety of spaces. So we can get subarachnoid CSF from a Sylvian fissure or from a sulcus. You can also get ventricular CSF. You can also get CSF from lumbar puncture versus a cranial. And I think few people have really looked to see whether that makes an impact or not. One of the first things that we've seen is that there's a significant difference between the cranial CSF proteome compared to the lumbar CSF proteome, even in the same patient. So these are paired cranial and lumbar samples that we've obtained at the same during the same operation. And we've seen that there's a far greater number of proteins that are more abundant in the cranial CSF than in lumbar. This is something that's now been mirrored in some of our data for D2-hydroxyglutarate for IDH mutant gliomas. We've seen the same thing for metabolomics or global metabolomics. Cell-free DNA as well works great in cranial CSF, half of the time in lumbar CSF. And this difference in cranial and lumbar CSF is so strong that it really doesn't even matter where you are in the cranial space. If you're in a ventricle, if you're in subarachnoid, even if you're in a resection cavity, the cranial to lumbar gradient is really conserved across all of those spaces. And why is this important? There were a couple proteins that for us had originally popped out, like fibroblast growth factor 1. Because we thought, well, if something is higher in cranial CSF, then it's closer to the tumor, and could it possibly tumor associated? FGF1 was one of the proteins that had popped out, but then we've recently started obtaining more cranial samples, even from patients with normal pressure hydrocephalus. And we see that FGF1 is also really elevated cranially compared to in lumbar CSF. And so I think FGF1 is probably just a protein of the cranial CSF space, independent of the disease that you have there. And importantly, when we're trying to think of what's a glioma-specific biomarker, I think it matters to compare things that are anatomically matched. This here is an example just of some proteins that I've pulled out, which have been significant whenever we compare them in cranial CSF from patients with gliomas versus NPH. But then if you look in lumbar CSF, again, from glioma and NPH, they're no longer significantly different. So thinking about where we are in location-wise does matter here. Similarly, it's not even just a question of cranial versus lumbar, but also a question of subarachnoid versus ventricular and also resection cavity. So this here are CSF samples from patients with GBM. And there was, again, a number of proteins that are specific to subarachnoid CSF, a number that are specific to ventricular CSF. One of the ones that'll pop out here is CAP-SL, which is a protein that's actually made specifically in the choroid plexus, which again would make sense why it's in ventricular CSF. And again, these are a lot of the work that we do is using training and validation cohorts or discovering validation. And whenever we split up our samples in half and look for these signatures, we see them reproducibly. But with that, we are really interested in trying to find monitoring biomarkers to turn that linear pathway that we were seeing before and get more information longitudinally from our patients. So the nice thing with an MI is that at least it's, sampling the same CSF location longitudinally, and so then location hopefully shouldn't matter as much unless you have changes with your resection cavity. But we've seen that there's a really significant difference between our pre-resection CSF and early post-resection from our patients here with gliomas. A lot of the pre-resection CSF proteins are enriched for these metabolic processes, whereas in early post-resection, we see that there's an increased abundance of more immunity-related pathways, which would make sense with recent resection. But we're interested in looking at these comparisons here because for us, a monitoring biomarker should decrease with resection, with cytoreduction, stay stable with stable disease, then also increase with progression. So one of the next questions then was, okay, so we know what goes down with resection. Well, what happens with chemoradiation? And, this for me was one of, at the time, one of the most exciting things we'd found so far, if only because it kind of went against what I thought chemoradiation did. So whenever we looked at our early post-resection versus our post-chemoradiation samples here, the post-chemoradiation samples were enriched for all of these neurodevelopmental, neurogenesis sort of pathways. And I remember, presenting this at one of our TAC meetings and saying, well, this doesn't make sense to me because radiation should just injure the brain. And one of my TAC committee members happens to be a radiation oncologist, and did not agree with that statement, but did ask, well, have you looked to see if you see a sort of similar proteomic signature if you don't, in patients who have not seen chemoradiation? So I looked, and indeed, this is just in one patient, but we've now seen this in more patients. In patients who haven't seen chemoradiation, the signature is very similar. And so I think what we're actually seeing here are the involving impacts of resection on the CSF proteome, and likely the reason why there's increased enrichment for these sort of neurogenesis-like pathways is because there's clearing out of post-resection debris that exposes what the signature of the brain looks like. So again, with that similar discovery and validation sort of processes here, but we've been able then to characterize proteins that are elevated in pre-resection CSF, a lot of which are metabolism associated. There's distinct proteins that are elevated in early post-resection CSF that then go down of delayed at a delayed time point. A lot of these are, again, these sort of immune-associated pathways. And then with the delayed post-resection, it's also tends to be a mix of some more brain-associated proteins, inflammatory-associated proteins. But, why is this important to us? I think one of the main things is that it's going to be, it's been challenging for us to deconvolute what's the impact of resection when we're trying to identify a protein that can be used for disease monitoring. Importantly as well, if you're doing a pharmacodynamic trial and you recently just had a patient who underwent resection, you give an immunotherapy and you look in this early postoperative phase, and you see that there's an increase in all these inflammatory associated proteins. Well, you might think my therapy is doing something, but is it really, or is it just the recent impact of resection that you're seeing? So I think we need to be careful here about what we're looking at, because this impact of resection we see even, at the time of recurrence, there's enrichment for these sort of resection-associated proteins. But nevertheless, we've still been able to find some really interesting proteins that we think may be useful for monitoring, like Brevican here, which is actually being evaluated right now under a CAR-T trial at UCSF. And it's increases at each time point with progression in this patient. We can also see, she had an EGFR amplified tumor, increase in EGFR over time. Some of these proteins then, decrease with stable disease, which correlates with what we've seen with D2-hydroxyglutarate if it's a patient that has an IDH mutant tumor. And importantly, despite ongoing pseudoprogression here in our patient 98, we then see also a decrease in these candidate monitoring proteins that again are congruent with the D2HD data that we're seeing, as well as cell-free DNA for this patient that's not shown here. And interestingly too, the CSF proteomics has been more difficult than any of us optimistically anticipated. But I do think there's a lot of utility and interest to it for both monitoring, but then also still for pharmacodynamics. So for example, here in our patient who had progressed through Avastin, the proteins that went up the most included VEGF-A, which would make sense with the Avastin, but then also different isoforms of VEGF, like VEGF-121, VEGF-165. So interesting questions there of potential resistance mechanisms. And then similarly here in a patient pre versus post pembrolizumab, we see a number of immune-associated proteins that went up, but including LAG3 as well, which is known to be potentially one of the resistance mechanisms to pembro, and is currently being evaluated through a number of trials using both anti-PD-1 and anti-LAG-3. So, number of conclusions so far, including the importance of looking at anatomical location and thinking about resection and other probably other factors that we're not yet considering when we're evaluating the CSF proteome. But I think there's a lot of promise to this, and one of our goals with this and with all of the data that we're generating has been to make these data publicly available for everyone to evaluate. So they're available with this QR code here. It's over about 147 CSF samples with more data to come that's under analysis right now. And we've also made all of our protocols publicly available with a recent review that was published by a group here that also details some of the potential challenges associated with liquid biopsy discovery and CSF, but then also some of the other promises that exist, not just with proteins, but also cell-free DNA and other things like that. So a lot of people to thank, but I'm happy to also take any questions now.
Wow, great work, Cecile. I'll maybe not ask questions because I'm a little bit more familiar with your work, but leave it to others to ask. So I should point out that although Cecile does work with me sometimes on clinical projects and things like that, her mentor for this work is Dr. Terry Burns, and I'll credit to him and to Cecile.
Wonderful. Yeah, amazing work, very impressive, and particularly building a biobank of CSF longitudinally and from different anatomical locations is quite valuable. So that's really great to see, and the results of that. For the audience members, if you have a question, I don't know if you're able to put questions from the audience. Feel free to raise your hand. I guess just to get started, I'll the question that comes to mind, and we've spoken a bit about this offline, but just sort of the logistics of getting longitudinal CSF is obviously very challenging, and something that your group has done a lot of groundwork to get off the ground. Can you maybe comment a bit about the logistics of all of this, and then also in terms of the effect of resection on the proteome? Are you seeing differences between sort of the ventricular or subarachnoid proteome versus the lumbar proteome? Like, is there less of an impact in the lumbar proteome after resection, or is it fairly similar?
Mm-hmm. So for the first question about logistics, I would say that the Amaya placement itself, intraoperatively is not that much of a challenge, but the logistical part then happens with being able to catch the patients at each appointment. And we're really lucky to have a team here of people who have volunteered their time very kindly to be able to tap these patients and to catch them at each of their appointments. We try to get the patients every single time they have an MRI. And then we also have been now getting longitudinal plasma from them as well. But honestly, when we first started, we were missing a number of the taps, the patient, the appointments weren't being scheduled and so forth. But we have some really great research coordinators who have been able to set up a notification system so that the appointment gets scheduled every single time. And then also it requires a large team of people being available to do the Amaya taps to be able to work around clinical schedules. For the impact of resection on cranial versus lumbar, I would say that's actually not something we have a really good answer for yet, in part because we are not really getting serial lumbar puncture samples from patients. I think the idea of getting an LP like every 1 to 3 months is not something most patients want. But I would, I'd anticipate that probably it would be diminished in the lumbar CSF. That said, I think you're working with less of a signal there to begin with anyways. So whether there's maybe more utility for lumbar CSF there. I'm not quite sure. We have looked at lumbar CSF for pharmacodynamics, so pre versus post pembrolizumab and NTI-7. I think we do see a signal there. It's just not as strong as the pre versus post in cranial CSF.
Thank you. Okay. I don't see any hands up, so maybe let me just double check. Yeah, okay, so maybe we'll close it out because we're at time. So just thank you, Dr. Parney, for being the first faculty discussant of this webinar series. We hope we'll have a couple more over the next few months. And thank you to the tumor section for hosting this, and particularly Dr. Haji Penayas and Dr. Dan Cahill and Dr. Gellerizadeh, who sort of spearheaded this initiative. And obviously, thank you to our three wonderful trainee presenters, Cecile, Adarsha, and Sanjeev. Dr. Parney and Dr. Haji Penayas, if you have any further comments, please go ahead.
No, thanks for the opportunity to participate in this. I think it was really great. I hope that we have more of these wonderful presentations, all three, and really some exciting work.
Wonderful. All right, thank
Thanks
you,
all.
everyone.